Good morning, dear student. In today's lecture, we will discuss the next important chapter of our course, that is transition metal complexes as chemical nucleus. In earlier lecture, we will discuss the chemistry of bismuth, but due to the unavailability of notes, we are discussing the chapter of bismuth in later on. So whatever the material which is available, I will try to give in the form of lectures. So so in today's lecture we are very much interested in the transition metal complexes as a chemical nucleages. So this chapter, it having a 4 lecture weightage. So uh, it include only 3 points, that is interaction of metal complexes with DNA, second, that is reactions of metal complexes with DNA, and the nucleus activity of this copper phenanthroline positively charged complex. So before discussing the interaction of metal complexes or metal ion with this DNA and RNA, we will discuss some introductory part, because uh, direct interactions of these metal complexes or metal ion with DNA, you can't understand before discussing some important points. At least you should know what is this nuclease, nucleotides, what are the components of these DNA and RNA molecules. So we are very much interested to study all these points, and then we discuss the remaining points of the chapter, that is the interactions and the reactions and the nucleus activity. So if we consider what is the heading of this chapter, that is transition metal complexes as a chemical nucleages. So what is this nucleages? So what is this nucleages? So why this transition metal complexes and metal ion act as a chemical nucleages? What are the function of these nucleages? Nucleages an enzyme. It is an enzyme that cleavage or cleaves the chain, that cleaves the chain of nucleotide, that cleaves the chain of nucleotide in the nucleic acid, in the nucleic acid. So these are the enzymes that break the chain of nucleotide in the nucleic acid. Nucleic acid, it is a word refer or stands for DNA and RNA. You should remember, whenever there is a nucleic acid, it is stands for DNA oblique RNA. So an enzyme that cleaves the chain of nucleotide in the nucleic acid. So what is this nucleotide? So let us discuss this second, that is nucleotide. So what is this nucleotide? It is an organic molecule. It's an organic molecule. Is an organic molecule that is the building blocks. That is the building blocks. The building blocks of DNA and RNA. So it is an organic molecule, that is the building blocks of this DNA and RNA molecule, made by 3 parts or 3 units. 3 parts or 3 units, such as phosphate group. One is phosphate group. The first one is phosphate group. Second, that is your 5 carbon sugar. 5 carbon sugar, it is known as pentose sugar. Pentose sugar. And the third one, that is the nitrogen bases. Nitrogen bases. So What is this nucleases? An enzyme that cleaves the chain of nucleotide in the nucleic acid. So nucleic acid is a polymer, and nucleotide is an organic molecule that is the building blocks of DNA and RNA, made up of by three parts. So one phosphate group, one sugar molecule, and one nitrogen bases. They combine, they gives one nucleotide molecule, and number of nucleotide molecules when interact, they will result into the polymeric structure called as nucleic acid, or we can consider it is as a DNA or RNA molecule. So you should remember, that is, the nucleotide is nothing but it is an organic molecule. uh, they are the building blocks of this DNA and RNA molecule. So nucleotide is a monomer, and this nucleic acid is a polymer, you should remember. So what is this nucleotide contain? It contains phosphate group, uh, 5 carbon sugar, it is called pentose sugar, and nitrogen bases. So we will discuss one by one, because this nucleotide is very important. So the phosphate group, it is very simple, and that is it is your P double bond O. This is your OH, O minus is there. Okay, this is your phosphate group, and it is attached to the sugar molecule, attached to the sugar molecule. So we are discussing the pentose sugar and the nitrogen bases. So the phosphate group, it is very simple as it is. So let's discuss the 5 carbon sugar. So you should remember this point, that is, a pentose sugar present in DNA, pentose sugar present in DNA, it is called deoxyribose sugar. And the sugar which is present in RNA, the pentose sugar present in RNA, it is called ribose sugar. DNA stands for deoxyribose. Deoxyribose nucleic acid, and RNA stands for ribose nucleic acid. So whatever the sugar which is present in the DNA molecule, it is known as deoxyribose sugar, and whatever the sugar which is present in RNA molecule, it is considered as a ribose sugar. So what is the difference? The difference between these two sugar is the presence of the hydroxyl group. So one of the sugar which is present in DNA molecule, it is considered as a deoxyribose sugar. If you consider in the deoxyribose sugar molecule, it is a pentose sugar, but one hydroxyl group is missing at carbon number 2nd. And if you consider this ribose sugar, then there is no absence of this hydroxyl group is present at carbon number 2nd. So if we discuss, let us consider a structure for this sugar. So if you consider this is your pentose sugar. So here OH, H, okay, here H, here H, OH, here H, this is your CS2, and this is your OH is there. If you consider numbering 1, 2, 3, 4, and this is carbon number 5. So it is your pentose sugar. If you consider at carbon number 2, there is no hydroxyl group. So hydroxyl group is missing at carbon number 2nd, and the sugar is considered as deoxyribose sugar. So one hydroxyl group is missing at carbon number 2nd, then it is considered as a deoxyribose sugar, and it is found in what? It is found in DNA molecule. This sugar, it is present in the DNA molecule. If you consider the second, the difference is only the presence of the hydroxyl group. So here HOH is there, HOH is there, here OH, H is there, H, this is your CH2 and OH is there. So at carbon number 2 hydroxyl group is present. So this is your hydroxyl group. Hydroxyl group is present. Hydroxyl group is present. So it is your ribose sugar. It is your ribose sugar. So you should remember, whenever a DNA molecule is there, there should be deoxy. So what is the meaning of this deoxy? Deoxy means without oxygen. So you should remember, deoxy means without oxygen. So at carbon number 2 one hydroxyl group is missing, and therefore it is considered as deoxyribose sugar. It is found in DNA molecule, while in ribose the second carbon it also contain one hydroxyl group, and therefore it is considered as a ribose sugar. When this sugar molecule, when it interact with, if you consider this oxygen, when it interact with phosphate group, and this oxygen is interact with the nitrogen base, then it is converted into nucleotides. So you should remember, nucleotide, it is made up of by 3 parts. One is pentose sugar, one is phosphate group, another is nitrogen bases. So this oxygen, when it bind with the phosphate group, and this oxygen, when it binds with the nitrogen bases, it will forms nucleotides. Number of nucleotides joined together to give nucleic acid, that is a DNA and RNA molecule. So this is regarding to the pentose sugar. Let us discuss the nitrogen bases of nucleotides. Nitrogen bases, it is also important. So if you consider, what is this nitrogen basis? A molecule that contain nitrogen, and it is properties like base, it is considered as a nitrogen base. If you consider the nitrogen bases found in RNA, DNA and RNA, so here a DNA is there, and here the RNA is there, and we can here write nitrogen bases. So which kind of basis is found in DNA and RNA? So if you consider, there is only one exception in DNA and RNA. All others nitrogen bases are similar. So if you consider DNA, the first nitrogen basis is adenine. It is denoted by symbol capital A. Second, that is guanine. Guanine, capital G. Then there is a thymine. Thymine, capital T. And the last one, that is cytosine. Cytosine. So you should remember, the nitrogen bases which is found in DNA molecule is adenine, guanine, thymine and cytosine. So if you consider this RNA molecule, again the adenine is there, second guanine is also there, so instead of thymine there is a uracil. So you should remember only these changes, and the cytosine is remain as it is. The only exception is that in DNA there is a thymine as a nitrogen base, and in RNA there is a uracil as a nitrogen base. So if we consider this adenine and guanine, so first two, this adenine and guanine, these are purines. Means they contain 6-membered as well as 5-membered ring. So this adenine and guanine, they are purines. They are 6-membered and they contain 6-membered and 5-membered ring, while the remainings, that is your thymine, cytosine and uracil. So thymine, cytosine and uracil, they are pyrimidines. means they contain only six membered ring. They contain only six membered ring. So uh, I will give you an assignment. So you should find out the structure of this nitrogen bases, that is adenine, guanine, thymine, cytosine, and uracil. Okay, we because in next lecture we will discuss the interaction of these nucleotides with metal ions and metal complexes. So at least you understand which nitrogen bases is there, which sugar is there, either it is deoxyribose or ribose sugar is there, and the phosphate group. So the phosphate group is very common, but which kind of sugar is there, either it is DNA molecule or RNA molecule, and which type of nitrogen basis is there, either it is adenine, guanine, thymine, cytosine, or uracil. So you should remember adenine and guanines are, they are purines. They are six membered and five membered rings, contain six membered and five membered ring. And thymine, cytosine, and uracil, they are pyrimidines, contain only six membered ring. So you should find out the structure of this nitrogen bases and try to uh, uh, draw the structure, because the structure are very slightly different, but you should understand the structure and try to draw the structure, practice it. In next lecture we will discuss the interaction, different types of interaction, that is covalent, non-covalent interactions of this metal ion and metal complexes with these DNA and RNA molecules. Thank you.